turn it over to Dr. Jennifer McQuade. We are really lucky to have Dr. McQuade here. She is an assistant professor of melanoma medical oncology, and I dare say a rising star, but actually an already established star. I think some of the really amazing work that Jen has done is in the setting of microbiome, and not only that, but you will hear about how even diet could be used as a therapeutic tool, which is kind of really being pioneered by Dr. McQuade. She is a physician scientist, and her lab is focused on proving that those approaches can be used in patients. The other thing I will mention is that Dr. McQuade has taken a special interest in survivorship and kind of the long-term impact of our therapies on our patients with melanoma, and I think very relevant to our melanoma brain metastases patients, given that, again, we're all very happy that now they live long and they will deal with some of these issues that Dr. McQuade and her team are focused on. So thank you so much for joining us, Dr. McQuade, and I will turn it over to you.
Great. Thank you, Hussein, for the introduction, and really thank you so much to AIM and to Sam for having me this morning and for putting on such a wonderful symposium. So I will get started. So what I'll be speaking about today, again, is not specific to brain metastases, but more about the role, what we're learning about the role of the microbiome. So these trillions and trillions of bacteria that live on us and in us, and diet in the setting of metastatic melanoma in general. And so these are my disclosures. And so as we've heard, we've made really tremendous progress in the treatment of melanoma in general, as well as of the treatment of patients with metastatic melanoma with brain metastases. And the unfortunate part, though, is that while many patients now benefit from these advances, we also know that some patients' tumors do not respond to our current therapies. And so the question really is, what determines response versus resistance? And you've heard some really elegant discussions, for example, by Dr. Davis about, what are some of these determinants of response and resistance? So, for example, the OX-PHOS pathway, et cetera. So we look at, are there things about the tumor cells that are different between patients whose tumors do or do not respond to immunotherapy? Are there differences in, say, the immune infiltrates into the microenvironment? Where I've actually really been interested in looking at is, we all know that we are different, right? Every single one of us is different. And we've learned from, for example, COVID, that there are certain features about humans that might make us either more vulnerable to an infection or have a different response. And so, for example, we know that, older and obese males tend to have more aggressive COVID, right? And are more likely to be hospitalized. Those are risk factors. And so we've known for a long time that some of these factors, such as things that we can't change, like age and our biological sex, as well as things that perhaps we can change, such as obesity, diet, exercise, and stress, these impact our immune system overall. Now, the question is, what is the impact of these factors on the immune system, specifically in the setting of our new checkpoint inhibitors, of these new immunotherapies? And that's a question that really had not been well studied. And one of the things that we're learning that makes this very, very relevant is the new role that we've established for the role of the microbiome. So again, these trillions of bacteria that live on us and in us, and how this actually impacts response to immunotherapy. And the microbiome is really interesting because it is actually an integrator of all of these factors. And we'll get into that a bit more. So again, what is the human microbiome? So the microbiome, we have trillions of bacteria that live on us and in us, kind of in a symbiotic relationship with us. They actually make up more DNA content than our own human cells. And what's really interesting about this microbiome and about this genetic content is, of course, we cannot change the DNA that we, were dealt from our parents. And we can't change the DNA of a tumor. We just want to eradicate the tumor. But the microbiome is actually largely determined by inputs and environmental factors. And so you will actually share more of your microbiome with, say, somebody that you live with that is unrelated to you than with your parents or your brother that live in a different state and have a different occupation and a different lifestyle. And so the microbiome has been well studied in the context of human disease, has been linked to obesity, to diabetes, to inflammatory diseases, to autoimmune diseases, and to the formation of some cancers, but had really not previously been studied in the context of melanoma, or specifically immunotherapy response. And the role of the microbiome was really first established in mice. And so these were two studies that were published back in 2015, basically showing that when we do mouse studies, what we do is we take mice of a specific genetic background, we give the mice tumors, and then we treat the mice with immunotherapy. And the goal is, of course, to learn, to develop more effective therapies, and also to learn about, what are mechanisms of resistance to our current therapies. Now, usually we know that it can be difficult and challenging to cure humans. And again, we're all different. In general, we say it's actually pretty easy to cure mice of cancer. Again, we're starting with something that's relatively simple this lab mouse, and we're giving them all the same cancer, and we're giving them all the same drug, which is different than cancers that develop within the human body. And so if we take the same, genetically identical mouse and we give that mouse chemotherapy, then, pretty much the mice usually kind of respond the same. Either, that particular tumor will or will not respond to the chemotherapy. It turns out that with immunotherapy, what happened is that we typically buy mice from two large centers in the US, either Jackson or Taconic. And you take the genetically identical mice and you buy them from one place versus another. And what it turned out is that the immunotherapy was only working in the mice from the Jackson lab and not from the Taconic lab. And so what they did is they thought, well, why are they different? It's the same genetically identical mouse. We're injecting them with the same tumor. What could be different? And so they profiled their microbiomes, these bacteria that lived in their gut. And what they found is that there was actually different microbiomes because they were being fed different food. They were living in different environments. And then if you actually did a fecal microbiome transplant from the Jackson lab mouse into the Taconic lab mouse, that you could actually transmit responsiveness to immunotherapy with this fecal microbiota transplant. And so the question, of course, became, is this relevant in humans? And so then investigators around the world, including my colleague Jennifer Wargo at MD Anderson, started collecting fecal microbiome specimens in our patients that were starting immunotherapy. And what we found is that there was actually distinct microbiome profiles between patients who did or did not respond to immunotherapy. Now, the microbiome is not everything. The microbiome is not the sole determinant. And again, there are plenty of patients that, have a good microbiome and their tumor does not respond. And there are other patients that might have what we would think is an unfavorable microbiome, and their tumor does respond. So there's different reasons for response and resistance, but the microbiome does appear to contribute. Now, we have a saying in science that correlation is not causation, meaning just because the microbiome is different between patients who do or did not respond doesn't necessarily mean that that is a microbiome that is leading to response or resistance. And so to get at causality, then this was again looked at in a mouse model. And so now taking fecal specimens from humans who either responded, so the top in blue, or did not respond to immunotherapy, doing a fecal microbiota transplant into a mouse and now injecting that mouse again with melanoma and treating them with immunotherapy. And what we found is that the mice who received the fecal specimen from the human responder to immunotherapy, these in blue, now their tumors shrink when they get immunotherapy. And if they got the non-responder fecal microbiota transplant, then their tumor would not respond to immunotherapy. So again, can transmit responsiveness to immunotherapy via the fecal microbiota transplant in mice. So the question, of course, is again, could we do this in humans? And there were actually 2 studies that were published a couple of years ago. And Erez Baruch is actually now a medical oncology fellow here at MD Anderson, works closely with Dr. Davis, actually. So there were 2 studies of fecal microbiota transplant in humans. And so in patients whose tumor had not responded to immunotherapy initially, so they had immunotherapy resistance. And then what they did is they did a fecal microbiota transplant from patients who had had a very good response to immunotherapy. And what they found is that in some patients now, with the combination of fecal microbiota transplant and then reintroducing them to immunotherapy, that they were actually then able to induce a response. Again, in some patients, not in everybody. Now, fecal microbiota transplant is, of course, a complicated procedure and is one that can only be done in certain centers. And it's, again, challenging to get those donor specimens. And so the question is, are there other ways actually that might be more scalable that we could use to modify the microbiome? And as I said initially, one of the things that's really unique about the microbiome is the fact that it is largely determined by external factors, by things that we can potentially modify. And so it's really not determined by our overall genetics. There's only less than 15% of it that's actually determined by our host genome. And it's largely determined actually by medication, so things that we ingest. And again, things that we ingest being the diet. And so with this, what we did is we started to integrate a lifestyle assessment into our microbiome collection protocol. And we published this work last year, essentially looking at 2 major factors. So the dietary fiber, and then whether or not patients were taking over-the-counter probiotics. And I'll show you some of this data. We also then took and did further mechanistic studies in mice. And so the bacteria that are associated with response to immunotherapy in our MD Anderson cohort are actually bacteria that we already knew quite a bit about from human health. We know that these are bacteria that have that help us digest fiber. So fiber, when we take it in, is what we call a prebiotic. It is actually a food source for the microbiome. And if we do not have those bugs in our gut, we actually cannot digest the fiber on our own. So again, it's kind of this symbiotic relationship. And we know that when these microbes break down fiber, that they produce these metabolites called short chain fatty acids, which are actually the primary nutrient source for the cells that line our gut and help us produce a nice thick mucosal layer that keeps the good bacteria healthy and helps us keep the bad bacteria kind of at bay. And so when we started asking our patients about about their diet, what we found is that our patients that had higher fiber intake in their regular diet, and again, no magic source of fiber here, but our patients that were eating more fruits and vegetables and beans and whole grains actually had an increased chance of having a good response to immunotherapy. Now, again, these patients might have been doing lots of other things, good too. And again, correlation is not equal causation. So we went to a mouse model, and now we take the mice and we feed them either the standard chow, which is actually a beautiful source of fiber, or we do fiber deprivation and inject the mice with melanoma again and treat them with immunotherapy. And what we see is when we change their diet, we can very rapidly shift their gut microbiome. And now if we fiber deprive the mice, what we find is that their tumors no longer respond to immunotherapy. And so the question is that can we intervene on the human microbiome through diet? So the data that I showed you before was about habitual diet. So what have patients been eating for a long time? The questions that my patients ask me is there anything else that I could be doing that might increase my chance of response to immunotherapy? So not what have you been eating, but what can I do now? And so there have been prior studies in non-cancer populations showing that if you swap diet, and these are pretty radical changes, that you can indeed modulate the microbiome. So this is a study where they took either African Americans living outside of Pittsburgh, eating kind of a prototypical Western diet, or Northern Africans with a very plant-based diet, and they swapped their diets and took them from basically less than 20 grams of fiber per day, which is what most Americans consume, all the way up to what the Northern Africans typically eat, which was more than 50 grams of fiber. And what we see in these graphs is that capacity of the microbiome to be able to produce these short chain fatty acids goes down, down, down as you're eating the hamburgers and French fries, and goes up, up, up as you're eating grains and legumes and plants. And then we have the opposite for meat metabolism. And so what we've been doing is now studying this in the context of melanoma patients, and specifically of patients receiving immunotherapy. We do this actually with what are called controlled feeding studies. So we all know, we all know, again, this is not a magic diet. This is the same diet that we should be eating for kind of overall health, for decreasing cardiovascular disease, diabetes, stroke, et cetera. But we know that diet change is hard. And what we really wanted to do was to, let's take out that behavioral component. And what we're going to do is we're going to actually give patients food. And when we give them the food, can we then change the microbiome? And so we started this with in kind of 3 phases, just like we do for drug discovery. So first with the phase 1 in our melanoma survivors, and then moving on now in patients that are actively receiving immunotherapy and looking, can we change the microbiome? And of course, if we do this, then the question is, if we change diet, can we improve the chance of response to immunotherapy? And there, we really have to start moving towards behavioral interventions, because of course we can't feed the entire world. And so what we see is that with this high fiber diet intervention, we try to ramp up our patients to between 30 and 50 grams of fiber daily, and patients are able to tolerate this. All of our patients were able to get up to that 35 to 50. This is during the diet intervention. And now afterwards, we don't ask our patients, actually, we don't give them instructions on how to continue this. And unfortunately, what we find is that people tend to go back to the same diet that they were eating before. And so it's an important point that we really need to learn how to better support people for long-term change. Our patients did eat the food that was provided. And when you ramp up the fiber that much, you get a little bit of GI distress at the very beginning, but it's all very, very mild and controllable. Now, we are not adding any new bacteria in. And so what we find is that we kind of shake up the microbiome within a person, rather than, again, introducing anything new. And our patients that have the most shakeup, not surprisingly, are those patients that started with a really fiber poor diet and kind of had the biggest change in their diet. We're also actually studying probiotic foods, so specifically fermented foods. And I'll come to why I'm talking about fermented foods and probiotic foods as opposed to probiotic supplements. But what we know is that these fermented foods, again, in other populations can help support the overall microbiome ecosystem. So not just the bacteria that are actually present within those fermented foods, but actually enriching the microbiome ecosystem overall. And this is in contrast to over-the-counter probiotics, which are typically single-strain probiotics, and that they're the bacteria that are easy to grow rather than necessarily the bacteria that are the ones that are the most beneficial. And so what we found is that our patients who were taking over-the-counter probiotics actually had a decreased odds of response to immunotherapy. Our patients that do the best are those that have high diet fiber intake and are not taking probiotics. And basically those over-the-counter probiotics are essentially crowding out some of the good microbiota, decrease the diversity of the microbiome, and now again in a mouse model actually diminish response to immunotherapy. I get asked all the time, what about after antibiotics? Should I take probiotics to actually restore the microbiome? And this was a study where they gave either humans or mice actually broad spectrum antibiotics, and then they looked at their microbiome. And what they found actually is that even with an 11 strain probiotic cocktail, that probiotic cocktail actually significantly delayed the recovery of the microbiome, again, because of competitive exclusion. So in conclusion, there is an intimate relationship between the gut microbiome and the immune system. And this also seems to be one of the factors that can influence response to immunotherapy. And we know now that the microbiome can be intervened upon. What can you do? Again, some of us do everything right and we still get cancer, or you do everything right in terms of diet, exercise, et cetera, and the cancer still doesn't respond. So these are not magic bullets, but again, what are some of the things that you can do that we certainly know will impact health overall? And the question is, now that we're trying to address this, does it actually influence response to immunotherapy? So no magic source of fiber, a variety of legumes, grains, vegetables, beans, increased probiotic food intake potentially, avoid unnecessary antibiotic exposure. Again, if you have an infection and you've got to take it, but avoid when possible, and avoid over-the-counter non-specific probiotics. And with that, I will turn it back over to Dr. Tobby.